Hello everyone and welcome to our presentation for Environmental Soil Microbiology SWAE3411. Today we will discuss the effect of cow manure on soil microbial communities. We are Group 4 and our team members are Ayman Al-Nadabi, Abdulaziz Al-Amri, Hamza Al-Hashmi, Hussam Al-Farsi, Abdurrahman Al-Awaid, Abdullah Al-Awaid and Jassim Al-Subayhi. In this study, we focus on how adding cow manure influences microbial abundance and the diversity in soil, especially bacteria, fungi, and actinomycetes, and what that means for soil health and sustainable agriculture. Let's start with a quick overview of what we will cover. Here's the outline of our presentation. First, we will introduce the topic and explain why soil microbes matter. Then, we will present our objective. After that, we will explain the methodology and materials used, followed by our results and discussion. Finally, we will end up with a clear conclusion and key takeaways. Now, let's move into the introduction and the scientific background. Soil is not just a dirt, it's a living ecosystem full of microorganisms that support plant growth and overall ecosystem stability. Cow manure is especially important because it can enrich soil by adding organic matter and microbial populations that increase biodiversity and improve soil structure. From a scientific perspective, we can study these microorganisms using microscopic and biochemical methods to understand their composition and activity in soil. Another major point is sustainable agriculture. When manure is used properly, it can reduce reliance on chemical fertilizers while maintaining or improving crop productivity. Finally, cow manure supports nutrient cycling. It contains microbes that help decompose organic matter and release essential nutrients that plants can absorb. So, overall, manure can support healthier soil by boosting microbial life and improving nutrient availability. So based on this background, we defined a clear objective for our experiment. The objective of our study was to identify the types of microorganisms present in a soil sample and to study how cow manure affects the distribution and abundance of these organisms in the soil. In simple terms, we wanted to compare untreated soil with treated soil and see whether manure increases microbial growth and whether it changes the variety of organisms we can detect, especially fungi, bacteria, and actinomycetes. Next, I'll explain how we designed the experiment and what steps we followed. The slide introduces our methodology. We followed a standard soil microbiology workflow. First, we prepared our soil samples and created a control and a treatment condition. Then, we performed microbial enumeration using serial dilutions and plating techniques to estimate colony-forming units, or CFUs. After counting colonies, we observed colony characteristics like texture, color, and shape to get an idea of microbial diversity. We also isolated pure cultures using streaking techniques, and finally, we used staining methods including gram staining and fungal wet mounts to support identification. Now, let's look in detail at the materials and preparation of our samples. For soil sample preparation, we used 2 cups, each of containing 150 grams of air-dried soil. 1 cup is served as the control, where soil was mixed with distilled water. The second cup was the treatment representing amended soil, and it included distilled water plus additional components such as 100% glucose, about 200 milligrams of ammonium nitrate, and elemental sulfur. These additions help stimulate microbial activity and make differences in growth easier to observe during incubation and planting. After preparation, both samples were processed using the same microbial enumeration methods to ensure the fair comparison. After preparing the samples, the next step was counting microorganisms through plating. To enumerate microorganisms, we began by preparing the appropriate agar media. Then, we created cereal soil suspensions, meaning we diluted the soil solution step by step to reduce cell concentration to countable levels. After that, we plated the diluted suspensions onto agar plates. This is accurate because if the sample is too concentrated, colonies grow together and can't be counted accurately. With correct dilutions, we get separated colonies, and each visible colony represents a colony-forming unit. An estimate of viable microorganisms in the sample. Once plates were incubated, we moved to observations and colony counting. After incubation, we observed microbial colonies on the plates and recorded visible differences. Then, we counted colonies using a colony counter device which improves accuracy and reduces human error compared to manual counting. At this stage, we focused on getting reliable CFU values for different microbial groups and comparing control versus treated soil. Next, to study organisms more clearly, we isolated single colonies using streaking. To obtain pure cultures, we used the streak plate method. First, we sterilized the loop using a flame until it became red hot and allowed it to cool. Then, we picked a small amount of sample and streaked the first quadrant of the agar plate. After that, we re-sterilized the loop and dragged from the first quadrant into the second, repeating the process for the third and fourth quadrants. This technique gradually dilutes the microorganisms across the plate, creating isolated colonies. Finally, we incubated the plate and selected individual colonies for further observation. After isolation, we used staining techniques to support identification. We performed drum staining for bacteria and wet mount for fungi. We prepared the slide, then applied crystal violet for 1 minute, rinsed with water, and added iodine solution for another 1 minute. We used ethanol for about 30 seconds to decolorize and finally counterstained with safranin for around 20 seconds. This process helps distinguish gram-positive from gram-negative bacteria based on cell wall structure. For fungi, the wet mount allowed us to observe fungal structures directly under the microscope. Now, let's look at the results, starting with CFU counts. This table shows the CFU results for different microbial groups in control versus treatment. Overall, we observed increases across all groups in the treated soils. For fungi, Set 1 increased from an average of 10.5 to 29.5, which is a large increase. Fungi Set 2 increased from 2.5 to 11, showing a moderate increase. For bacteria, BYA3 increased from 7 to 17, which is a significant, while BYA4 increased slightly from 1 to 3. For actinomycete, GCA3 rose strongly from 3.5 to 18, and GCA4 increased from 0.1 to 1.1. Results suggest that the treatment condition supported more microbial growth and higher abundance. Counts are important, but morphology also tells us about diversity. So, let's examine colony traits. Here, we summarize microbial morphology by group, media, colony traits, and likely types. For bacteria grown on PYA media, colonies were described as smooth, round, creamy, and moist. This morphology can be consistent with gram-negative bacteria such as Pseudomonas or E. coli. For actinomycetes, on GCA media, colonies appeared chalky, leathery, and filamentous. This is typical of streptomyces, which are gram-positive and known for their filament-like growth. For fungi, on rose Bengal media, colonies were cottony, velvety, or powdery with white, green, or yellow colors. These features often match common soil fungi like Aspergillus, Penicillium, and Trichoderma. So, beyond increased counts, we also see signs of more diverse microbial community. Now, we can summarize what these findings mean for the effect of cow manure on soil health. This slide summarizes the overall effects observed. First, microbial abundance increased, shown by higher CFUs across all groups. Second, colony morphology indicated greater diversity and complexity in treated samples, included a mixture of gram-negative bacteria, gram-positive bacteria, actinomycetes, and different fungi. So, overall conclusion from our observations that cow manure or manure-based treatment can enhance microbial activity and support improved soil health. This is important because microbial diversity is strongly linked to better nutrient cycling and more stable soil ecosystems. Finally, let's conclude with the key takeaway points. To conclude, cow manure treatment led to a clear improvement in soil indicators. First, we observed increased microbial abundance, meaning higher CFU counts of bacteria, fungi, and actinomycetes in treated soils. Second, the community appeared more diverse, supported by colony morphology and staining observation, showing gram-negative bacteria, gram-positive bacteria, and actinomycetes, and also fungi. And third, these changes suggest sustainable benefits. Manure supports nutrient cycling, decomposition, and long-term soil fertility, helping reduce dependence on chemical fertilizers. Overall, our results support the idea that organic amendments can improve soil biological health. Thank you for your listening. Now, we welcome your questions.